First, I would like to thank Hervé and Muriel for inviting me to this meeting, for all of you for being here. And I think this is a very good opportunity to all of us to learn more about astrobiology. Yes, I'm Carlos Briones. I work in the Center for Astrobiologia, Hervé, Astrobiologia in Spanish. So, astrobiology, exobiologie, as you prefer, in Spain, and I'm the responsible of the molecular evolution group there. I'll be talking this morning about early life, simple life. This is the title Muriel suggested for this morning. So the first part will be delivered by me, and the second one by David Moreira. This is the overall picture of astrobiology. You have seen this picture many times, and we are supposed to be interested in all the events from the origin and evolution of the universe, the formation of planetary systems, the interaction among the atmosphere, lithosphere and hydrosphere in some of these planets, the origin of chemistry that is complexified progressively until it acquires the capacity to self-copy, to mutate, to evolve, and at the end to give rise to the wonderful biodiversity that surrounds us. Today, as I told you, we are going to focus on this part of the overall picture, so the origin of life. Origin of life has a lot of unsolved questions. When, how, was it the effect of chance and or necessity? Early means simple. Where, in particular, terrestrial origin of extraterrestrial origin? Did it originate once or several times? Okay. This picture kind of recapitulates the main events we have been discussing the previous days. So the origin of the Earth and Moon was dated 4.57 giga-year or billion years ago. First rocks and crust and oceans were present at 4.4. The Earth, all the sedimentary rocks are probably 3.9. The isotopic fingerprints of early life, for sure, let's say, are dated back 3.85, but perhaps 4.1, and the oldest fossils are in this range, okay? So the origin of life is something that somehow happened in this time period. Probably here some of the chemicals start interacting and reacting also with the extraterrestrial matter delivered by comets and meteorites, and after the late heavy bombardment, perhaps the life had more opportunities to start and start developing. Okay. After this part of the history of our planet, evolution, I will not tell a single word about the evolution, about this part of the picture, which is fascinating, as you can imagine. But we will focus on this origin of life, okay? There are two complementary approaches, as you know. The first one is to study or to try to approach the origin of life from a bottom-up perspective, also called from past to present, and from chemistry to present, so from the non-living. There are many disciplines that use this approach, in particular astrochemistry, geochemistry, prebiotic chemistry, and more recently synthetic chemistry. Okay, and the complementary one is the top-down vision. Okay, so from present to past. So from the living, from the current biodiversity, comparing the current biodiversity to the non-living. This is the field of paleontology, microbiology, phylogenetics, and minimal cell models. Okay, and here you have this yellow box, what is in fact a black box of the origin of life. Okay, so I will combine somehow these two approaches during my talk, and probably David will concentrate in the top-down approach. We can imagine our planet being something like this picture, 3.8 billion years ago. Lots of energy sources, Probably interaction between the water, this planet was plenty of water at this time, and the rocks there, and the atmosphere. So, in fact, our planet was a kind of giant laboratory of chemistry, okay? Different micro or macro environments were there. It was possible to synthesize some compounds in the atmosphere because some gases were present there. Also, as I told you, extraterrestrial delivery of organic matter was evident at that time because lots of meteorites and cometary nuclei were falling to the Earth. Interfacial chemistry between the surface and the water. Of course, aqueous phase chemistry. We are not sure about the temperature of water at that time, but okay, the range here is quite generous. My friend Jean Clips wrote from 0 to 100, so okay, probably. So liquid water. We don't know about the pH of that water, the amount of reagents, we will tell something about that, and the concentration of them. And also hydrothermal and geochemical synthesis at the bottom of the oceans. Okay, so probably all this was interacting, all this was providing different environments for chemistry to evolve or to start reacting. In this setting, the oldest fossils, I just want to recall these ideas, that the first stromatolites are probably, who knows, these ones that resemble microbial mats. We were talking about this yesterday. It's quite controversial if this older rock is also an stromatolite, as we were discussing the previous days. We have this famous picture. Yesterday we were talking a lot about soft microfossils. It's not clear by any means that these are all cyanobacteria. And the Brasier group also provided nice evidences about quite old microfossils. But as Puri told us yesterday, it's possible to produce in the lab without any involvement of biology biomorphs, and this is the work of our colleague Juanma García Ruiz in Granada in Spain, that closely resemble some of the microfossils. So, danger. So, be careful. So, morphology is not a biosignature of life. And this also applies to this recent discovery. Okay? These are hematite tubes. Okay, this is by the way, this is a reconstruction, a very nice reconstruction of the data. But are they originated by microbes? We don't know. Okay, but this is the time scale from, let's say, 3.8, 3.9 or 4 until 3.5. Something happened there, and chemistry turned to be biology. Let's focus a little bit on the composition of life. As most of you know, just this, all of you know this is a periodic table, and these are the elements that life uses. Okay, so 99% of our bodies, of all life, all the bacteria were in our body and in the environment of the birds, are made of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. And the remaining 1% is distributed as you can see here in this periodic table. Okay? This is quite different from the composition of the stars and planets. As you can see here, stars are mainly composed by oxygen, composed of oxygen and helium, and this 2% hydrogen, sorry, hydrogen. I was looking at all the picture at the same time. Hydrogen and helium, of course, and the remaining 2% are the so-called metals by astronomers. For us being chemists, okay, carbon is a metal, okay, but 2% of the stars are not hydrogen, not helium. The crust of our planet is mainly silicates, so oxygen, silicon, aluminium, iron, and something else, but the living beings, we are mainly oxygen, carbon, and hydrogen, and also nitrogen, phosphorus, and sulfur, and the other 1-2% of the elements I showed you before. As an example, this is a typical textbook example. Our body is composed of 65% oxygen, 18% carbon, 9% hydrogen, and 3% nitrogen. So it's quite easy from a chemical point of view to construct living beings. This is so, this composition of living beings, because we are mainly water. We are surrounded by water. The surface of our planet, mainly here in this area, is covered by water. And as we saw the previous days, water is indeed the best solvent we can imagine. It allows chemical reactions. It also provides thermal stability, which is essential for life. And it's very interesting because it's not only a solvent, but it plays an active role in molecular processes related to life. Okay? And also, it's a very abundant molecule in the universe, so no problem in that sense. And the other main component of life from a chemical point of view is carbon, as you know. Carbon is quite peculiar because it allows a complex chemistry based on covalent bonding, so sharing electrons. And different carbon-carbon structures can be formed, both aliphatic and aromatic. And it's also important because it allows producing or forming functional groups with the other 5 main elements of life. If we go further in the complexity of life, some small molecules are essential for us. A bit more complex molecules that are present in all organisms as cofactor, coenzymes, whatever. Then the monomers of the macromolecules of life, so nucleotides, amino acids, sugars, and fatty acids. And then the main molecules of life, the polymers, DNA, RNA, and proteins, okay? And you know that in biology, DNA replicates thanks to some proteins that perform this function. Then it is transcribed into the RNA and then translated to proteins in the ribosome. So this is the main flow of genetic information in life, and this is quite important for life to evolve and to maintain. And also viruses are able to replicate RNA and also to retrotranscribe RNA into DNA. So this would be the overall picture of the genetic flow in our biosphere, okay? Then the next step in complexity are cells, as you know. All life is based on cells, as Puri told us yesterday. 2 main organizations of cells, prokaryotic and eukaryotic, with a difference you know. Prokaryotic cells belong to the domains bacteria and archaea, and eukaryotic cells, eukarya. Okay, it's basic biology, basic, let's say, biochemistry. But what is life? Life is something more than the composition of life. It's more than the sum of the parts of life. This is a very interesting question that has been faced since the beginning of our culture, and this is a scheme borrowed by David and Puri. In this year, they were so patient to take some of the main definitions of life since Aristotle to now. Okay, we don't have time to go into details, but here you will see the number of biologists, from philosophers, from physicists, from chemists. So all of us are interested in trying to define life. I don't know if it's worthwhile or not to define life, as we were discussing the previous day. But I think that it helps us in the sense that it can allow us to try to dissect the main features of life, okay? So if you try to define something, you pick up the main features of that thing you are trying to define, okay? In recent definitions of life, it's evident that life requires these 3 main components: a replication system, a genetic molecule that stores and transmits information; a metabolism, a set of reactions that are used to interchange matter and energy between the living being and the surrounding; and a compartment, a membrane, let's say, in current biology, that allows this metabolism to be performed, okay? Because this is an open system that allows an out of equilibrium chemistry inside the compartment, okay? So the combination of replication, metabolism, and compartment make possible the appearance of life, okay? So these are key features in life. The combination of these three features is not present in non-living materials, okay? So, having this in mind, I just want to underline three definitions, recent definitions of life that I like more than others. Christian de Duve in 1999 said that life is a system that can maintain itself in a state far from equilibrium, this is important, and that can grow and multiply with the help of a continuous flow of energy and matter from the environment. Okay? It's a quite nice definition of life. Then Jerry Joyce said that life is a self-sustaining chemical system capable of undergoing Darwinian evolution. And this very short definition of life was adopted by NASA Astrobiology Institute as an operational definition of life. This is good enough to distinguish from non-living to living beings. And also, my colleague Kepar Ruiz-Mirazo and others recently wrote that living beings are autonomous systems with open-ended evolution capacity. So as you can see here, apart from this combination of compartment metabolism and genetics, the evolution is important in the definitions of life. Why? Because as you know, nothing makes sense in biology except in the light of evolution. This is one of our mantras in biology, okay? So, something similar to this is life, okay? David will probably tell us a lot about phylogenetic trees, about the reconstruction of the history of life from a top-down approach. So, lots of trees will probably appear in David's talk, but I would like to concentrate on the roots of the tree, because the origin of life is something that happened here, using this metaphor of the tree of life, okay? Then the diversification of life there is another issue, okay? The first scientific ideas, because I don't have time to talk about spontaneous generation. That is a very interesting history that was refuted by Pasteur, but this wonderful French scientist in the 19th century. But the first scientific ideas about the origin of life were written by the great Charles Darwin. Okay? This is Charles Darwin on a petri dish. So, microbiologists are quite freaky sometimes. Okay, so this is Charles Darwin, and at the end of The Origin of a Species, this wonderful book that you should read at least once in your life, the last paragraph says, "There is grandeur in this view of life, with its several powers having been originally bred into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful have been and are being evolved." This is a very nice sentence. And he says, having been originally breathed, this is the first edition, only breathed, not by the creator, as in the second one, breathed into a few forms or into one. Okay? Then some years later, in a letter he wrote to his friend Hooker, he said, "But if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc., present, that a protein compound was chemically formed, ready to undergo still more complex changes, at the present day such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed." Okay, so the warm little pond, some compounds. This sentence is somehow the origin of the top-down approach to the origin of life, and this sentence is somehow the origin of the bottom-up approach to the origin of life. So again, Darwin was there. Okay. Then the first model were proposed by these two great scientists of the beginning of the 20th century, Alexander Ivanovich Oparin and Jean B.S. Haldane. This one working in Russia and this one working in UK. There are some similarities and differences, but I would like you to see the main features of the Oparin's model, the heterotrophic origin of life. Okay, so he was favoring a reducing atmosphere in the planet, where the synthesis of organic compounds could have been possible, and then the formation of the so-called primitive soup, formation of coacervates, groups of proteins and other things together, and then the primordial heterotrophs, so the first living beings. This was published first in Russian and then in English. The first book, this one in Russian, The Origin of Life, it's quite clear, 1924, okay? And then Haldane some years later. 30 years later, the first experiments showed up. They were performed by Stanley Miller in 1953, as most of you know, and also this Spanish scientist Joan Oro, okay, and at the end of the 50s. And these first experiments turning the problem of the origin of life from, let's say, the library to the lab. That was done 30 years after, in the 50s, and it was the origin of the prebiotic chemistry as a scientific field. Okay, this is the famous Miller experiment. Miller was favoring the Oparin's view of the earliest atmosphere, and he decided to mix in a flask 4 gases that were supposed to have formed the primitive atmosphere, very reduced forms of oxygen, hydrogen, nitrogen, and carbon. Okay, 60,000 volts were applied here, so this was the energy source that would resemble the sparks and the fall of meteorites and so on primitive Earth. And 2 weeks later, lots of amino acids and other biomonomers appeared. Okay, so this was the first experiment of prebiotic chemistry. So chemistry itself has the capacity to produce the first building blocks of life. Of course, nowadays we think that this atmosphere was not so reducing, and in particular, CO and CO2 were present in the early atmosphere of the Earth. But okay, the experiment was relevant itself. It was also very important because some years after Miller's experiment, a meteorite, a carbonaceous chondrite, similar to the Allende meteorite we were able to touch a couple of days ago, fell in Murchison in Australia, and okay, carbonaceous chondrites have lots of organic material. When this material was analyzed, the part of organics showed that it contained carboxylic acids, the dicarboxylic amino acids, hamides, and other compounds. And if you compare the composition of amino acids, in particular the aliphatic amino acids, you compare their presence in this Murchison meteorite or Allende meteorite with those that can be synthesized in different kinds of atmospheres using Miller-type experiments, you see that the composition and the relative abundance is quite similar. So perhaps Miller-type experiments were performed elsewhere, and extraterrestrial delivery of perhaps amino acids could be present at the origin of life. So this was relevant. The other experiments of these pioneers were performed by Joan Oró, as I told you. He was able to combine 5 molecules of hydrogen cyanide to form adenine, so the first nucleobase that could have constituted somehow nucleotides and nucleic acids. Joan Oró was quite famous in the States, where he was working at that time, and also in Spain. And our former king, Juan Carlos Briones, named him the Marquess of Oró. And he had to choose his coat of arms, because if you are named a Marquess, you have to choose the coat of arms. And he decided to put in the coat of arms the stars, his wife and daughters, a mountain, typical mountain in Catalonia, and adenine. So this is the first biomolecule present in a coat of arms in the history of heraldry, which is quite interesting. Okay, so these are the pioneers. 60 years, no, not 60 years, some years later, and I'm only giving some clues about prebiotic chemistry. We don't have time to talk about prebiotic chemistry in deep. But it's also interesting that the chemototrophic theory suggested an alternative origin of life based on the assimilation of CO2 and the formation of acetyl-CoA, long molecular weight compounds, macromolecules, and primitive cells. This is a complementary view to this heterotrophic origin of life. There are 2 pathways for early carbon fixation, the reverse citric acid cycle and this pathway. This is biochemistry. I don't want to bore you with this data, but I think it's interesting for you to know this. This was the first of these. Also, he was also a pioneer, Günther Wachter-Heuser, and this kind of chemistry is associated with hydrothermal vents. Okay, and 60 years later, nowadays, prebiotic chemistry is a bit more complicated, because scientists have been able to suggest, to model and to perform different kinds of reactions that originate different biomolecules to define the environment that could have accommodated those reactions. And okay, this is a quite complex and interesting view nowadays. And one open issue is, of course, the composition of that primordial soap. What kind of organics were there? Every scientist has its own recipe for the primordial soap. Some of them favor very diluted one, only water, some oil, a limited number of compounds. Others prefer more condensed soups, in particular containing traces of minerals that can catalyze processes on the surface. In fact, this surface-based catalysis is very important. It's more and more recognized as a key in the origin of life. So the model of a soup is being modified, and perhaps it was a pizza, primordial pizza. And what is very important is the chemistry you can accommodate in the interlayers of some minerals. For instance, clay, montmorillonite, so interlayer, so primordial lasagna. Who knows? But these scenarios, these metaphors of the scenarios are currently being considered in laboratories, and who knows where life originated. Okay? In particular, it's also interesting this. The primordial soap was endogenous. I mean, the composition, the ingredients were from all of them, on Earth or in Earth, depending. Or, as we were discussing yesterday, some of the ingredients could have been delivered by meteorites and cometary nuclei. We don't know, but this happened. So probably this very complex laboratory at 3.8 or something like that billion years ago contained both endogenous and extraterrestrial organics. Okay? The point is that you can have a plethora of different ingredients, of different organics, but it's very difficult to try to imagine how this mixture could be somehow selected chemically selected to form the first monomers and biopolymers of life. And this is nowadays one of the open issues in this field. Okay? In any case, from the precursors that were present on our planet, okay, using all we know about prebiotic chemistry, biomonomers could have been produced, and this is the roots of the tree of life. Okay, here you see Luca, bacteria, archaea, and eukarya, and this is the trunk, the common trunk of life. But okay, you have to go up and you have to reach Luca. What happened here? We don't know. But somehow DNA, RNA, proteins, membranes should have originated. Okay? So the major transitions in the origin of life could be starting from physico-chemical conditions present on early Earth and going to Luca. Here you see the evolution of life. Okay? Here, fossils cannot tell you anything about that, because fossils only cells fossilize, not molecules, and this is the field of molecules. This was a molecular world before the first living beings, or depending how you define life, these complex systems harboring metabolism, compartment, and genetics. Here, and then Luca, and then evolution. So fossils cannot help you here. Molecules should have self-organized, then a genome and replication appeared, and a membrane and metabolism system, and these two in combination. Okay? Because otherwise, if you have only this or only this, you don't have life. So we use, again, bottom-up and top-down approaches. Okay? So this is the field of origin of life nowadays. And there are, as you can imagine, lots of controversies in this field. I have listed here only 3, and we can discuss later, or as we have suggested, Muriel, after David's talk, if you are interested, we can have an open discussion with Puri, David, and myself to try to give some clues or ideas or whatever about this origin of life related stuff. So one of the controversies is genetics first versus metabolism first. Okay? Those favoring genetics, okay, claim that it's very important to have coded information and the appearance of the first self-replicative systems. But metabolism first says, okay, without bioenergetic mechanisms, you don't have life. You don't have this conversion of matter and energy. Recently, and I will concentrate on this in the second part of my talk, some systems approaches have showed up that try to combine protometabolism and genetics. Protometabolism in the context of a compartment scenario. So reproduction of the whole system. That would be probably more real, more biological. Other controversy, heterotrophic or autotrophic earliest metabolism. Probably David will tell us something about that. Heterotrophic origin. Okay, the primordial soup provides organic molecule and energy. Okay? You go to the supermarket, you take their molecules, you eat them, and that's all. But for this to be possible, okay, you need a complex prebiotic chemistry, because you need lots of different molecules. And then a simple metabolism, relatively simple metabolism can be performed if you have a bit of everything around. And then the chemoautotrophic origin from CO and CO2 to cells. So you take a carbon from CO and CO2, and you use, in most cases, geochemical energy sources. In this framework, a simple prebiotic scenario is possible. You don't need so many organic molecules around, but you need a quite complex metabolism. This is an open issue, how complex that metabolism could have been. But this is an open issue in the field. And also, early protocellular or mineral compartments for life. Membrane-based compartments, okay, allow bioenergetics and reproduction, because you can facilitate the flow of matter and energy through across the membrane. But mineral surfaces are rigid, or they are related to chemotrophic metabolism. So perhaps in this version of the earliest metabolism, this kind of compartments are favored, and for this, that one would be the best option. Nowadays, all life, and in Lucas you will see, for sure, we had membranes. So this was the winner of the possible two compartments available. Okay, so these are three of the many controversies in the field of origins of life. And also it's very interesting, something that Puri also showed us yesterday, that okay, I told that okay, this is a kind of a physical-chemical scenario, perfect. You have here the chemistry of the earth, Luca, the trunk of life, diversification of life, but you could have different origins. Okay, why not? Nothing impedes that at this stage different mechanisms of combining molecules and trying to get a system able to reproduce itself would be present. Perhaps some of them required some organic material that was absent at a given time, or perhaps a meteorite fell there and destroyed one small little pond with this small tree of life. We don't know. And at the end, we are the descendant only of the only origin that survived. So as Puri showed yesterday, and I have chosen the same picture because it belongs to a chapter Puri coordinated in this book. Here you see the tree of life, and these, okay, are possible origins, alternative origins that disappeared, and here you will have Luca and current biodiversity. Okay, that's why we usually prefer to say origins of life than origin of life. Okay. The second part of my talk, I will focus on the RNA world. The RNA world is one of these models about the origin of life. You saw lots of controversies, so there are different models, but RNA world is one based on this genetics first scenario that has been very popular for the last 4 decades. Okay. Okay. First, basic. DNA versus the RNA. You know these two molecules. This is the typical double helix of DNA. This is the monomer. We call it the deoxyribonucleotide. And this is RNA, usually single-stranded. Okay, ribonucleotide. And these are the 4 nucleobases for DNA, 4 nucleobases for RNA. So uracil instead of thymine in RNA. And also here in the monomer you see an OH group, hydroxyl group here, which is lacking here. Okay, so from a compositional point of view, this is the difference between DNA and RNA. It's important to recall that nucleobases, these molecules, are always interacting to each other through complementary hydrogen bonding. Okay, so A and T, or G and C, okay, or G and U in the case of RNA. Okay, let's focus on RNA. RNA has a sequence, of course, A, G, C, U. You can follow the sequence here, but in solution, all RNA molecules fold, okay, and establish secondary structures based on this hydrogen bonding, as I told you. And different kind of secondary structure elements appear. Here you have a stem, a loop, a bulge, an interior loop. We biochemists are very fond of these kind of things. Okay, then the interactions become more complex, and for instance, this loop can interact with this one if they have complementary nucleobases. So tertiary interaction appear, and at the end, the structure of RNA in solution is quite complex. It's by far more complex than that of DNA, which is a double helix and nothing else. Okay, so you have the sequence of the molecule, And as I will show you, it can take genetic information in some current replicative entities and the structure that can perform biochemical functions as proteins do. Okay? So this is the chemical basis of the pleiotropy of functions that RNA shows. Indeed, something which is interesting, but I don't have time to go into detail, is that there is a degeneration between the space of sequences, the possible number of sequences you can have, and the possible number of structures. Because, for instance, here, all these sequences will look for a complementary base pairing and will fold into the same structure. Okay? And what performs function is the structure. So you can have different sequences that are performing the same function. So you can accommodate lots of mutations here and the function will not be affected. This is very important in the case of RNA. So this degeneration, okay, gives RNA robustness and adaptability. And also it's interesting, and it works so wonderful. This nice GIF shows how an RNA molecule is in solution trying to find the most stable structure, okay, and complementary base pairing is playing there, and at the end all the structures that a given sequence can form can be ordered by the free energy, so the most stable will be probably present there longer than the less stable ones. So RNA is, okay, very plastic molecule in that sense. Native structure of RNA can be visualized because here you see only secondary structure, planar representations of RNA, but for instance in my lab we have used atomic force microscopy, okay, to visualize individual molecules of RNA on a surface in native conditions, so in water, okay, or in some buffer we use. And here you see a nice transition between a very elongated shape to a very compact one, depending on the amount of magnesium you have in the medium. Okay, so RNA, the structure of RNA is a key issue, okay, in the biology of this molecule. Then, flow of genetic information. I will give you some clues about RNA, and then I will try to put all of them together. You see this. Replication of DNA, transcription and translation. In some viruses, replication and retrotranscription. Okay, DNA, RNA, protein, perfect. RNA is in the middle, good. As a central molecule. When we concentrate on the translation of genetic information, so the conversion from messenger RNA into proteins, we let's say we were amazed by this particle. This is a ribosome. It's probably the most fascinating macromolecular assembly in the cell. These are the large subunit. This is the small subunit. The sequences Puri was telling about yesterday come from here. These are RNA, and all these small objects here, the dark blue and the purple, are proteins. Okay, mRNA, messenger RNA, goes here. This is the transfer RNA, and this is the nascent peptide, and at the end it will exit the ribosome as a folded protein. Okay, so this is wonderful. we now precise information about the structure of ribosomes. And as you see here, most of the ribosome is RNA. In particular, here, where transfer RNA puts together the different amino acids, in this region, there are no proteins. So RNA is performing the so-called peptidyl transferase function, so joining two amino acids. So it's essential for the flow of genetic information, for the conversion from a RNA-based dictionary to a protein-based one. Okay? Also, lots of regulatory mechanisms have been discovered in bacteria since these very earlier publications, and also in eukarya. So RNA is also a player in the regulation of the flow of genetic information, and also it's a genetic molecule. Okay, RNA is a genetic molecule. It carries genetic information in viruses. Here you have the famous Baltimore classification of viruses, and in all these families the genetic material is RNA. Okay, double-stranded, single-stranded, one polarity, the other polarity, combinations of them, so lots of options. But RNA is the genetic material in a lot of the viruses, in lots of these replicative entities. I don't call them living beings, replicative entities. In fact, if we focus on the main families infecting vertebrates, okay, here you see the kind of genome they have. Double-stranded DNA, single-stranded DNA, single and double-stranded DNA, for instance, hepatitis B, and double-stranded and single-stranded RNA. So everything is possible in the viral world. Here you have very nice examples, well, nice if you are not infecting you, nice examples of RNA-based viruses. Okay, so RNA is acting as genetic material nowadays. And if you analyze how viruses, RNA viruses replicate, you see that they, I don't have time to go into details of this, but they are something like this. For instance, if some of you have been infected by a virus, of course all of us are infected by a RNA virus nowadays, for instance common cold, probably, or perhaps other kind of viruses, the distribution of the genomes of the viruses in our body is something like this. All viruses are different to each other. We can have 10 to the 12th, 10 to the 13th different viruses in our body. And when they replicate, they originate a population that is also very different. So it's impossible, unless you perform a bottleneck, to have an idea of the mutations you have distributed in the different genomes in the consensus sequence. Okay, this is called a quasi-species. This is a kind of population that allows us explaining how viruses evolve nowadays, and that could be relevant for having an idea on how RNA molecules in RNA wool could have been organized in heterogeneous populations. Okay. Also, RNA is the genetic material in viroids. I don't know if you have heard about viroids, probably only biologists. These are plant pathogens where the infectious entity is only an RNA molecule, no capsid, no viral structure, only an RNA molecule. It's naked, it's circular. Naked means without proteins around. It's short, compact structure, and what is important is that it doesn't code for any protein. So RNA is the genotype and the phenotype. This provides a template for replication, and there are two families. And this is an example. The numbers are very weird in most of viruses. Potato spindle tuber viroid, because this is the effect that the viroid produces. These are normal potatoes, regular potatoes, and these potatoes were infected by this viroid. Okay, and this is the replicative entity, an RNA molecule. So the genome of a viroid is an RNA molecule. That's all. Okay. Indeed, it's quite interesting to trace the relationship between the mutation rate of different replicative entities and the genome size. Okay. Here, the group of Ricardo Flores is using this not so accurate higher and lower eukaryotes, but in any case, it's inversely proportional. Okay. And viroids are the replicative entities that accumulate more mutations when they replicate. Then RNA viruses, and at the end, some eukaryotes. More proofs in favor of RNA. RNA is able to catalyze biochemical functions. Here, again, the flow of genetic information, replication of DNA, transcription of RNA, translation of RNA into proteins. This is the ribosome, and this nice picture shows different amino acids here, and this is the protein. Okay, good. This is a picture in eukaryotes. It's quite similar in bacteria and archaea, but this is a bit more complicated. If you focused on this part, you can ask yourself, okay, all the messenger RNA is going to be translated into proteins? The answer is not. Most of our genes, of the genes in nature, have two parts, exons and introns. Introns are DNA regions that are represented in messenger RNA and that will be represented in the final protein, okay? But introns are DNA regions that are not present in mRNA. The term splicing was coded to name this reaction of intron excision and intron ligation, something like this here. This is a gene. You have exon, 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 and introns. Introns, in principle, are not relevant for the final protein, okay, a simplification, and they are spliced. They are eliminated from there, and this is, okay, how mRNA is, let's say, presented to the ribosome to produce the protein, okay? The splicing of RNA is basic in the evolution. For instance, it allows the alternative splicing, so because you can splice this or not, or this or not, so different mRNAs can be coded in a single DNA sequence, complex sequence. So this is basic for the synthesis of immunoglobulins in vertebrates. Okay. And people were studying splicing, and groups of proteins and RNA combined were able to cut the introns and join the exons. And okay, they were very happy with this kind of pictures. Exon, exon, intron, ribonucleoproteins here, and the proteins cutting this end, and then cutting this one and joining this exon, and the splicing is produced. Perfect. But science is always very, very interesting. And in 1980, in Boulder, Colorado, in Tom Check's lab, they were studying Tetrahymena thermophila, which is a unicellular eukaryote. And one of the genes of this Tetrahymena had one exon, intron, exon, okay? And they say, okay, let's do some molecular biology. Let's take this gene, let's take the nucleus of the cell, extract the proteins of this nucleus. In the proteins, some of the proteins will be responsible of the splicing. So if you take part of the nuclear structure of this and mixed with the gene, the splicing process will be produced. And okay, we put it in a test tube. We like very much test tubes in molecular biology, as you know, with some salts that are required for the process, in particular magnesium, and an energy source, GTP. Okay? And an experiment, and one gel. Okay. And this is very interesting for the molecular biologists here in the room. In the three cases, the gene, okay, was added to the reaction. The nuclear extract was added in lanes 1 and 2, and magnesium and GTP in 1 and 3. Here, the first one with everything, okay, you have the long form of the gene and the spliced form. Perfect. If you don't have magnesium, if you don't have GTP, splicing is not possible. And if you don't have nuclear extract, the splicing is produced. But the proteins responsible for the splicing were here. So what happened? So why this band was here? How was it possible to splice an exon in the absence of proteins? As you can imagine, in Tom Check's lab, this experiment was repeated a number of times to be sure that that was the outcome of the reaction, and at the end that was the outcome of the reaction. So mRNA splicing can be performed in the absence of proteins. That was revolutionary because it showed that it is possible for RNA to auto-catalyze its own escission and ligation. So RNA is performing biochemical functions, and they code the name ribozyme for this kind of enzymes of RNA. Okay? And this was the original paper on this, that, by the way, was rejected from Nature and Science because it was too revolutionary for that time. Okay? Okay, after that, they were able to propose the mechanism of self-splicing, and here you see the G from GTP somehow cuts here, and in the presence of magnesium this 3' end also cuts here, and at the end this is joined, ligated. This is possible because the intron, this part, I mean the part of the molecule that apparently was not important, had a 3D structure that allowed all this biochemistry to be performed and allowed this escission and this ligation. Okay? If you want to see this intron in a more friendly view, this is. Okay? So this is the first ribozyme that was discovered. So this is an enzyme made of RNA. So this works as current enzymes, and all living beings have ribozymes inside. Okay? After this discovery, a second type of self-splicing introns were discovered. They are longer, more complex. I don't have time to go into details, but they are also performing self-splicing. And almost simultaneously, the group of Altman was working in the ribonucleoprotein, so a complex of protein and RNA, that is responsible to cut this end of the transfer RNA. This 5' end must be cut. And okay, the idea was that the ribonucleoprotein that cuts this end, composed by protein and RNA, the catalytic function was performed by the protein, because that was the idea at that time. But they were able to discover that it is the RNA of this complex which performs the catalytic function. So it was the second example of ribozyme. Okay? And it was published 1 year later, okay? 1983. Since then, 8 different natural ribozymes have been discovered. Here you have some of them. 7, these 4 represent other 3. 7 of the ribozymes catalyze the excision and ligation of RNA. So this is the biochemical function they perform. And the 8th one is peptidyl transferase, that, as I told you, is a ribozyme inside a ribosome that catalyzes the joining of amino acids. Also, other ribozymes, lots of them have been produced by in vitro evolution of nucleic acids, as I will show you later. Okay? So this is also something that favors the functionality of RNA. This is so, and Tom Check, the discoverer of ribozymes, wrote a very nice review some years later, and he suggested that perhaps at the beginning you can have ribozymes or ribozymes combined with peptides. These peptides not produced by translation, so not biologically produced, but produced by abiotic means. For instance, you can have surfaces that catalyze the polymerization of amino acids into short peptides, and this can perform catalytic functions. In some cases, these ribonucleoprotein enzymes are stable and perform very well, and so on. And later on, most of the catalysis was, let's say, focused on protein enzymes, but some ribonucleoproteins exist nowadays where the catalytic function is performed by RNA. Okay? Let's move to in vitro evolution of RNA. Okay. After the discovery of the structure of DNA, the double helix and so on, Crick and Orgel and others, and Crick and Orgel and others, said, okay, this is DNA, RNA, proteins, but what if RNA was the first genetic material in the history of life? That was very, let's say, very complicated, because it was the DNA era. But okay, they in the 60s they started thinking about that, and there was one researcher that asked himself, okay, could it be possible? I mean, if they say that RNA could have been the first genetic material, could it be possible to show the evolution in the test tube in the absence of cells, in the absence of viruses? So could RNA molecules somehow evolve by themselves? Okay, this is the main player of this story, Sol Spiegelman, a very smart scientist, as you can see here, and this is the original article in the 67. What he did is he took a virus that infects bacteria, the bacteriophage Q beta. He took their genetic material, this long, relatively long genome, 4,000 nucleotides long, and he said, okay, if I put some of this genome in a test tube and I add some of the molecules that the RNA genome needs to replicate, so a protein, RNA replicates, and some salts and so on, I would expect that the RNA replicates and accumulates, and the concentration of RNA in the test tube will increase until the ribonucleotides and salts are exhausted. So I need to transfer it to a new tube with more of this to, okay, again and again, allow the replication, the protein-based replication of RNA. Good. He did it, and it was okay. It was okay, as expected. The amount of RNA in the test tubes was increasing. But at the same time, they observed that a shorter version of this genome appeared and replicated very, very fast. And the shorter version of these molecules was shorter and shorter and shorter and shorter until only about 200 nucleotides. So the amount of DNA that the protein needs to start replication. So the protein, okay, replicated the whole genome and also shorter versions, okay, and they replicated faster because the selective pressure there was replicate fast, because you are going to be transferred to a new test tube, okay. So this new molecule that showed up was called Spiegelman monster, because it was the first nucleic acid molecule produced in the lab artificially that was able to be amplified. Okay, so this was the beginning of this line of research of in vitro experimental evolution. But it was very difficult to perform this kind of experiments, and let's say that biochemistry was not so developed by that time. It was about 23 years later where these two groups, the group of Larry Gold and Jack Szostak, developed a system called systematic evolution of ligands by exponential enrichment that allowed the in vitro evolution of nucleic acids. How this works? Okay, you can imagine that you start with an initial population of RNA molecules, a large one, say 10 to the 13 different molecules in solution, okay? You can imagine here, this room, plenty of molecules, okay? You can be molecules if you want. And you are folded. Now you are folded, okay? You are folded in solution. RNA molecules are folded. And the selective pressure introduced by the researcher is, okay, you have to be able to bind, for instance, this molecule. Okay, you have molecules like this that cannot bind, but some of them can bind it. Because in the population of 10 to the 13 molecules, some of them, by chance, will have a more or less, let's say, optimized or pre-optimized pocket to accommodate this molecule. Okay. You can, if you fix this, you bind this ligand to a surface, whatever, you can be able to say, okay, those binding the molecule are good for my experiment, I keep them, and I discard the remaining molecules. Okay? These RNA molecules that were selected because they were able to bind this ligand can be retrotranscribed to cDNA, PCR amplified to have more copies of DNA, and then transcribed again to have a new population of molecules. A new population that has been previously selected by its capacity to bind the ligand. Okay? If you repeat and repeat and repeat and repeat this process, at the end you will have a populations of molecules, all of them or most of them being able to bind this ligand. So here, this is the evolution in the test tube. Okay? You have amplification and selection. Okay. This is a wonderful means for proving how evolution works, and also for selecting functional molecules with the applications that you are interested in biochemistry, in medicine, in environmental control, whatever. Okay? We are playing this in the lab. And for instance, this is examples of aptamers that we have raised against some molecules of a virus. And you see here the secondary structures of these aptamers, and they are very good in binding these molecules. Here you see other nice examples of aptamers. This is interesting, this collection, because this was selected against adenosine monophosphate and adenosine triphosphate. Okay? Here the aptamer is the large part of the picture, and this is the ligand. Okay? And you can see how the evolution in the test tube has allowed to select a very good pockets to accommodate the ligand. And here in this case, the ligand is a protein, so larger than the aptamer. Here, the ligand is this, and the aptamer managed to enter in one of the holes of the protein and be very it increased the affinity very much for this protein. There are also molecules similar to aptamers in nature, but I will not tell you anything about that. So, recapitulating, if we come back to this DNA, RNA, protein model and say, okay, what came first, DNA or proteins? Because proteins are required for this. The ribosome made up of proteins and RNA are required for this. So this is the chicken and egg paradox, okay? But if we put in a list the properties we have been discussing, okay, the information can be stored in DNA and in RNA, viruses and viroids. The structure is mainly the responsibility of proteins in living beings, but RNA is also part of the structure, for instance, in the ribosome. And, very important, catalytic function. Proteins, of course, enzymes, but ribozymes. So RNA can perform catalytic function. So, at the end, what came first? DNA or proteins? Perhaps RNA. This is the basis of the RNA model. Okay, this is why I wanted to show all these pieces of the recent history of molecular biology. There are more proofs that favor the antiquity of RNA versus DNA, and also this is important because most of the molecules living beings use as energy coins, GTP, ATP, are ribonucleotides, so can be derived from that RNA wood. And also in the metabolism, deoxyribonucleotides, so the precursors of DNA, are always the outcome of a reaction performed on ribonucleotides. So our metabolism is somehow showing that RNA was very important at the beginning. and it's very difficult to think about proteins being here before RNA, because protein biosynthesis is dependent on RNA, as you saw, and on ribozymes. So the RNA model says, okay, RNA is the only known macromolecule that can function as genotype and phenotype, sequence, structure and function, the molecule itself, thus allowing evolutionary processes at the molecular level. This is something RNA can do. And okay, if this is true, an RNA world could have existed as an intermediate step between prebiotic chemistry and current DNA-based life. Okay? This model, okay, would, let's say, accommodate in the overall, like this, more or less. Perhaps at the beginning, RNA was replicating itself and performed catalytic activities. Then proteins appeared because peptidyltransferase ribozymes started to code proteins, a very simplified explanation, and then DNA RNA protein and the current world. So nowadays RNA is an intermediate molecule, but perhaps at the beginning it was the main player of the molecular evolution world. The model of the RNA wall was published by Walter Gilbert in 1986, and here he wrote, one can contemplate an RNA world containing only RNA molecules that serve to catalyze the synthesis of themselves. Okay, some years later we think that this only is too much. Probably the current versions of the RNA world must take into account that the heterogeneous scenarios that was present at the origin of life at this intermediate step between prebiotic chemistry and LUCA contained peptides, cofactors, low molecular weight compounds, inorganic catalysts that could somehow help RNA in this RNA world. And this is the current version of the RNA world. Not RNA only, but RNA plus other small molecules. Not protein, nor DNA, but small molecules. Okay? Indeed, experimental protein cells are being designed nowadays. This Jack Szostak is doing a very nice work in Harvard, and also David Deamer. And okay, steps are being taken towards the production of the so-called ribocyte, replicative entity that could be able to reproduce itself based on RNA. We are far from that goal, but very interesting work has been published during the last years. Again, let's go back here, and okay, if RNA world was correct, this picture could somehow fill the gap between prebiotic chemistry and LUCA. But there are some problems, of course. Is it possible the prebiotic synthesis of RNA? How can we produce the first RNA molecules that were the main players of the RNA world? I don't have time to go into details, but there were some problems in the past to be able to produce in the lab this bond, in particular, among the sugar of the ribonucleotide and the nucleobase. But fortunately, the work of John Sutherland in 1999, this is a commentary of Jack Szostak in the same issue, showed an alternative pathway to produce ribonucleotides. So this was the traditional one. Okay, you have here some small molecules that, okay, researchers try to combine to get activated ribonucleotides that could be polymerized to form RNA, but this was not working, and this was very inefficient in this sense. Sutherland was clever enough to suggest an alternative mechanism using this intermediate molecule that, okay, doesn't use this part of the pathway and produces activated ribonucleotides. This is a very interesting approach, because we are biased by how we see molecules nowadays. If we see a molecule with a phosphate, sugar, and nucleobase, we say, okay, the traditional, the easiest way would say, okay, let's produce this, this, and this, and then join. Perhaps nature did something very different, as here is exemplified. And this was the beginning of the systems view. So perhaps you can play like a chemist instead of playing like a biochemist if you are going to try to explain some steps of the origin of life. Play with all the molecules you have around and try to get the final results. Okay? The problem is the polymerization of ribonucleotides. Once you have, let's say that with these alternative pathways, published by Jean in 2009, okay, you can produce the ribonucleotides, but then you have to polymerize them to get RNA. You need activation of ribonucleotides. In nature, ribonucleotides are activated by 3-phosphate group. In the lab, usually imidazole is used as an activator molecule because the polymerization of ribonucleotides is favored with this group. But in any case, in bulk aqueous solution, it was shown that it's very difficult to polymerize RNA, because if you have two activated nucleotides and you want to produce a dinucleotide, the reaction is not favored at all. Okay? So different strategies have been developed to reverse the direction. First, thermodynamics, using water-poor media, okay, and using environments that promote nucleotide stacking, that could favor the process of joining ribonucleotides, and also using kinetics to improve the biopolymerization rates. In that sense, without going into details, but mineral surfaces are being fundamental to allow the polymerization of nucleotides and to form up to 50 mers of RNA. So, lasagna again, okay? Lipid bilayers, because the interface between lipids and water media can concentrate molecules and somehow promote the polymerization of ribonucleotides. And eutectic phases in waterized media, because again it concentrates the reagents and promotes the polymerization. So you see, things are improving. Then another open issue is, okay, I imagine that I can polymerize RNA using, for instance, mica here. This is montmorillonite, and this is an activated nucleotide, and Ferris and others showed that, okay, you have one nucleotide, and this is an electrophoresis gel, up to 40 mers, 50 mers in a montmorillonite enriched media. Okay, but you have to try to go to these kind of molecules that are currently being proposed as the first molecules that could have been able to catalyze the replication of other RNA molecules, including themselves. So if this is construct and evolved in the lab, this is not present in nature, okay? But if these molecules were able to replicate a copy of themselves, and so being the first RNA-based RNA polymerase, okay, from 40 mers to this, it's not so easy. We have developed models, for instance, that can fill the gap somehow, and we have shown that you can end with a RNA polymerase using ligation-based strategy, because among the initial repertoire of oligonucleotides you get from abiotic polymerization of RNA, number of them have this structure, a hairpin structure, one of the possible structures, and part of these hairpin structures can be ribozymes, can be hairpin ribozymes, as I showed you before. So, without going into details, you can have oligomers, polymers, monomers, let's say 40 long. Some of them are ligases that can ligate and make the puzzle and end up in a RNA-dependent RNA polymerase. Okay. But in any case, was RNA the first genetic polymer? We saw, we also hear this before here. No, probably not. Probably other analog molecules lacking ribose and in other cases lacking ribose and phosphate could have been present at the interface between prebiotic chemistry and an RNA world, and this is a very active field of research nowadays. And in particular, I show you the structure of RNA and PNA. If PNA, peptide nucleic acid, here you see the backbone of this molecule is more similar to a protein than to a nucleic acid. If something like this is artificial, okay, but if something like this existed in the past, perhaps it was easier to polymerize than RNA, and somehow transferred the genetic information to an RNA, and then RNA took the main role. Okay, PNA and RNA fold in double helixes, so this kind of artificial molecules, artificial nucleic acids, are very interesting also in this field of the origin of life. So, summing up, if this is prebiotic chemistry, this is a possible timeline. Okay, perhaps pre-RNA world were there, then an RNA world on the first ribocytes. Okay, RNA protein world, DNA RNA protein world. Okay, we could talk about this for hours, but I think this picture is good enough for you to have an idea of the overall process. What about viruses? We can discuss later on if they should be considered as living beings or not. But perhaps the first viruses appeared once you had RNA-based cells, and perhaps the first DNA viruses appear once you had DNA-based cells. And from that time, viruses have been appearing lots of time. Okay? But in any case, as Darwin said, this is what we think, what scientists think. Nothing in this picture is true. Is what we think about that. And I will finish in 5 minutes. Oh, okay, I've been talking for a while. Okay, I will finish in David, sorry, our idea was to talk less than that, but okay. Couple of slides. RNA, in any case, as I told you, should be considered as part of the overall number of processes that accumulated from the prebiotic world and the first self-replicative entities. Because remember, for a system to be alive, it's required that it has a metabolic machinery, compartment, and genetic template mechanisms that gives the system autonomy, individuality, and history. These are three main features of living beings. And as you can imagine, all this RNA world stuff, all this molecular biology work that I am performing in the lab, and lots of researchers are performing in the lab, is an oversimplification of the reality, because this is our experimental setup. Here you can have molecules of RNA replicating or whatever, but okay, you have the genetic and template mechanisms. You are providing the compartment, the test tube of the researcher, and you are providing the metabolic machinery, because you need to add proteins and enzymes, something previously formed by the metabolism. So the point is, okay, let's deepen into the possible origin of metabolic machinery, compartment, and genetic template mechanisms as separate parts of the picture, or better, as common, interrelated, interconnected parts of the picture. This is the idea of the prebiotic systems chemistry, this new field that is now quite popular in the field. Because as Ernst Satzmari wrote some years ago, you need the combination of these three components. You can think about designing in the lab binary systems, but at the end you have to go to the ternary systems if you want to produce living beings. The first relevant step in that sense has been performed again by John Sutherland in his lab in UK. They used these precursor molecules that in the presence of UV light and hydrogen sulfide, again, hydrogen sulfide appear sometimes this week as reductant, and you can use also this photoredox cycling. You produce somehow the main molecules that can form the compartments, or lipids, the main molecules that can be combined to construct the genetic and template mechanisms, so nucleotides, and the main players of metabolic machinery that are amino acids that then can be combined into proteins. And they even suggest a prebiotic scenario based on impacts that is quite interesting, because it is connecting the chemistry in the lab with the real, okay? So perhaps this kind of warm little ponds or this kind of impact craters were relevant because they allowed the mixture of molecules here, of complex mixtures of molecules coming from elsewhere, to combine to produce complex enough media that were required for life. And we're working in this kind of stuff, which is quite complicated, but what is important, and I would like to underline only this idea, is that you have to try to link replication of the genetic material with the reproduction of the system, because this is how life works. So the coupling between the genetic control of metabolism that is able to take a molecule and construct the compartment in a way that this complex system can reproduce, this is the key issue nowadays. This is very difficult to implement experimentally, and this is some of the kind of work that synthetic biology is doing in the bottom-up approach. But in any case, as John Sutherland published very recently, we are far from the origin of life. We are somewhere here, okay? He says that we are at the end of the beginning. It's a very nice metaphor. Something happened here. Perhaps the threshold between necessity and chance, contingency, was somewhere here, and then a long, long way to look at. Okay? So we have to be very realistic at that point. Okay? I would like to finish with this picture again. Darwin told us that the chemistry probably in the primordial, in the early life, early Earth, was very complex. We have to have that in mind, because life started on Earth, not in the test tube of a researcher. As Jacques Sostak used in the title of one recent paper, an optimal degree of physical and chemical heterogeneity, complexity, is needed for life to appear. And I had the opportunity to be with Jacques Sostak, John Sutherland, and others a couple of months ago in Madrid in a meeting we organized. And when discussing about experiments related to the origin of life, John told me, let's make it dirty. Let's make it complex. Let's make it real. Thank you very much.